Good morning. This is our last revision and our last session of biology. I'll be revising the main concepts or reviewing the main concepts on this PowerPoint presentation for your semester 2 test. The first question is about aerobic organisms, anaerobic organisms, and facultative aerobic organisms. But let me first see the title and remind you of the title of the PowerPoint presentation, which is the cardiovascular system. So I might ask you a very simple question at the beginning, which is, what does the cardiovascular system consist of? So the cardiovascular system simply consists of the heart. Cardio refers to heart, and the blood vessels. Vascular refers to the blood vessels, and the blood vessels are the arteries, the veins, and the blood capillaries. Now, coming back to aerobic organisms. Aerobic organisms are organisms that can respire only in the presence of oxygen, meaning that they can produce energy mainly or only in the presence of oxygen. Whereas anaerobic organisms, obligate anaerobic organisms, are organisms that can respire only in the absence of oxygen. That's to say, they can produce energy only in the absence of energy. Of course, all human beings are aerobic organisms and are obligate aerobic organisms. But there are some tissues in the human body that can respire anaerobically if they do not receive sufficient amounts of oxygen, like the muscles, for example, and in which case they would undergo an anaerobic process called acidic fermentation, and in that process they produce lactic acid, and lactic acid causes pain. And lactic acid is the reason why we call this type of anaerobic respiration acidic fermentation. Now, what's the importance of getting energy? Energy is important for vital processes inside the living cell or inside the living organism. For example, active transport processes, active transport of sodium, for example, or potassium against concentration gradient from lower to higher concentration requires energy. Similarly, all active processes that require energy, like thinking, for example, like moving, all these are active processes that require energy. So if you are an aerobic organism, then comes the next question: what's the importance of oxygen for aerobic organisms? Why is oxygen so important? Or you might face the question or encounter the question in a different wording. I might ask, what happens if aerobic organisms are deprived of oxygen? Or a third form of the question, why does deprivation of oxygen cause death in aerobic organisms? This is because oxygen is important for breaking down the bonds that store the energy in large organic complex molecules like glucose, where oxygen acts as the final acceptor of electrons. So if I ever ask you, what would happen if oxygen is insufficient or oxygen is completely absent, then there wouldn't be an electron acceptor, and therefore there wouldn't be breaking down of bonds, and therefore there wouldn't be a release of energy that is needed for the vital processes, and the cell would die or the organism would die. Another question is, why do higher organisms require a transport system? Higher organisms are made of complex, trillions of complex of cells, and therefore there are some superficial cells on the surface, and there are other cells that are deep in the central part of the organism. Simple diffusion is enough for transport of gases, for example, from the outside environment to the superficial cells. But simple diffusion would take a huge time or a huge period of time to allow such gases to reach the central part of the complex organism. Therefore, it is much easier and faster to have a transport system with a pump and transport vessels in order for these substances to reach the deep parts of the cells or the deep cells in a short period of time. Another question is about hemoglobin and the need for hemoglobin, or justify the importance of hemoglobin in the human body. Hemoglobin is a pigment, as you know. Why is it a pigment? Because it contains certain chemicals and iron salts that make it have the red color. But the importance of hemoglobin is not in being a pigment. The importance of hemoglobin is in being a respiratory pigment, meaning that it's a pigment molecule that helps in respiration. And as I said before several times, there is a difference between respiration and breathing. Breathing includes the breathing movements, the mechanical breathing, inhalation and exhalation. But this is not at all respiration. And that's why we always emphasize that we have to say cellular respiration, because cellular respiration is something that occurs at the level of the cell, inside the cell. What is cellular respiration? It's nothing but the breakdown of the bonds of glucose or any carbohydrate in order to obtain energy. So how can hemoglobin help in respiration, and why is it called a respiratory pigment? Because the structure of hemoglobin as a protein allows for active sites for the hemoglobin molecule to bind with 4 oxygen molecules. So one hemoglobin molecule will bind with 4 oxygen molecules. And since hemoglobin is contained in the red blood corpuscles, and since red blood corpuscles are carried by the current of blood to the cells, therefore hemoglobin will reach the cells carrying oxygen. And fortunately, there is a very important structure function adaptation here, is that the binding between hemoglobin and oxygen is a loose binding. Is this something good? Here it is something positive. Why? Because this looseness of the bond will allow hemoglobin to release oxygen easily at the tissues and to carry to be bonded to carbon dioxide in exchange. Now, having known that oxygen is being carried by hemoglobin, you might be asked another question, which is, how is oxygen transported in blood? When you're asked such a question, how is oxygen transported in blood, you have to notice first that the beginning of the question is how, meaning that you have to explain the way oxygen is being carried in blood. So actually, whether we are talking about oxygen or carbon dioxide, both or each of them is carried inside the RBCs bonded to hemoglobin, as well as dissolved in plasma. But there is a big difference between the two, because if you're talking about oxygen, then the major part of the oxygen carried in blood, about 97 to 98% of that blood, is carried on hemoglobin forming or bonded to hemoglobin forming oxyhemoglobin, whereas only 2 to 3% of the oxygen in blood is carried in dissolved in the water of plasma. On the other hand, for carbon dioxide, it's exactly the opposite, but not with the same percentage. The majority of carbon dioxide is dissolved in plasma in the form of carbonic acid or bicarbonate salts, and only a minority of carbon dioxide is bonded as it is, as carbon dioxide, to hemoglobin, forming carbaminohemoglobin. You needn't remember the word carbaminohemoglobin, but I'm just saying it because it's not carboxyhemoglobin. Carboxyhemoglobin is something different, okay? And now we need to mention you have a famous question you've frequently encountered on the test, which is the structure function adaptation of the red blood corpuscles. Of course, as you see on the diagram, the red blood corpuscles are very small in size. They are microspheres. The diameter of the red blood corpuscle is about 8 microns. It's true that they are bigger than the body cells, but at the end of the day, they are still very small to be able to pass through the small capillaries. If they were big, they would be stuck in the small blood capillaries around the cells. Number two, they are not completely spherical. They are biconcave. And this biconcavity means that they are concave on both surfaces, the upper surface and the lower surface. And this increases the surface area for diffusion of gases in and out, whether oxygen or carbon dioxide. Number three, they are devoid of nuclei. They cannot divide, they don't have nuclei, and the absence of the nucleus is good for the red blood corpuscle because it allows it to contain or to accommodate more hemoglobin. The fourth structure function adaptation of the red blood corpuscle is that it has a very thin, flexible membrane, and that, again, the thin membrane allows it allows for a better diffusion process, and the flexibility of the membrane allows it to be intruded in the small blood capillaries. Now, inside the blood, of course, you know that blood consists of plasma and cellular components. You know that the cellular components are the red blood corpuscles, which we call corpuscles because they are spherical and non-nucleated, and to differentiate them from other cells which are nucleated, we have the white blood cells that are of different types, and we have the blood platelets. Let's talk first about the platelets. What's the importance of the platelets? First of all, platelets are not complete cells. Platelets are fragments of cells, meaning that you have a big cell that's called the megakaryocyte. You needn't remember that word. And that is fragmented in the bone marrow to give rise to small non-cellular fragments, each of them being called a platelet. As long as the blood vessels are intact, the platelets are carried in the blood smoothly without any interference. But once there is injury in the blood vessel, an injury in some of the platelets start to secrete a sticky substance, and that sticky substance helps the platelets adhere together, and in addition to other cellular components like the red blood corpuscles and the white blood cells, as you see, and at the end of the day, you would have a thrombus or a clot, meaning that the blood would be converted into a semi-solid substance at the site of the vascular injury or the injury of the blood vessel, and therefore the process is called thrombosis, but the clot itself is called a thrombus. And as you see, the thrombus would be very efficient in plugging the site of the injury and therefore stopping. In the blood, you also have white blood cells, and you know that white blood cells are important for the specific immune specific defense mechanisms of the body that are called immunity. And we have two types of, or we have several types, of course, of white blood cells that are involved in non-specific and specific defense mechanisms. but let's focus more on the ones that are concerned with specific defense mechanisms or immunity. They're called the lymphocytes. They have a very particular shape. They do not have a multi-segmented nucleus like this one, but they have a central, rounded, very characteristic nucleus. And those lymphocytes are divided into B lymphocytes and T lymphocytes. Both are important for specific immune mechanisms. The B lymphocyte by secreting antibodies that make an antigen-antibody reaction with the antigen, with the ultimate result of destroying that antigen, whereas we have another type of lymphocytes that are called the T lymphocytes. They do not secrete antibodies like the B lymphocytes, but they secrete cytokines or chemicals that result in a direct destruction of the cell that carries the antigen. So they are more, T lymphocytes are more concerned with the direct killing of the foreign cells. Why do we need transport vessels? We said that we need a transport system, and therefore, of course, we have to have transport vessels, because a transport system is nothing but a pump and vessels. Why are the vessels that important? The vessels are important, first of all, because they contain the blood. We want to make that blood reach the tissues, and we want to make that blood that carries oxygen and nutrients reach the tissues. Not only do we want it to reach the tissues, but we want it to reach the tissues at a convenient rate that satisfies the needs of the cells of oxygen and nutrients. And therefore, the vessels contain the blood. The pump allows the blood to be moved in such vessels at a suitable rate. But we will be learning again that the vessels themselves, with their elasticity, and the muscles in the wall, they help the blood reach the tissues. And moreover, there are very specific blood vessels that are called the arterioles, that are the end vessels in the arterial system. And these arterioles have smooth muscles in the wall. The arterioles are very important because they have a very important and significant function in the cardiovascular system, which is to fine-tune or to control the volume of blood that reaches the tissues, or to control the blood flow to the tissues. How do they do this? How are they adapted to this? By having smooth muscles. So if I ever ask you about the function of the arterioles or about the significance of the arterioles, you would talk about fine-tuning or regulating the blood flow to the tissues. And you will be starting by smooth muscles and ending by regulation of the blood flow. And in between them, there will be steps. So if they have smooth muscles in the wall, these smooth muscles can contract or relax, and therefore the whole arteriole can constrict or dilate, and this is how they regulate the blood flow to the tissues. In between the arterioles, which are the end station in the arterial system, and the venules, which are the first station in the arterial, in the venous system, we have the venules. And in between the arterioles and the venules, we have the blood capillaries. The blood capillaries are extremely important. Number one, because they are very fine blood vessels that surround the cells. So they help the blood reach every cell in the human body. They are very thin-walled. They are numerous. They are branched. They surround the cells from everywhere. They have fenestration. And these fenestrations allow the leakage of plasma, or the exudation of fluid, carrying oxygen and nutrient to the tissues, and then carrying back carbon dioxide and waste substances from the tissues. So First of all, they are thin-walled. Their wall is made of only one layer of endothelial cells. Number two, they have fenestrations. Number three, they are numerous in number, and therefore, although they are very fine vessels, in order to be able to pass in between the cells, yet they have a huge surface area. All this allows exchange of material between the blood and the surrounding cells in tissue capillary exchange, or between the blood and the air sacs in alveolo-capillary exchange. So they are made of one layer of cells. They do not contain, I forgot to say this, they do not contain any muscles. They have fenestrations, they have a very large surface area, and all this will help the process of diffusion on both sides, or exchange of material between the inside, the blood, and the outside, which depends upon whether you're talking about capillary tissue exchange or capillary alveolar exchange. So let's first describe the capillary tissue exchange. In capillary tissue exchange, carbon dioxide, at the arteriolar end, oxygen and nutrients, will diffuse from the inside of the blood to the tissues. And carbon dioxide and waste substances will diffuse from the tissues to the inside of the blood at the venular end. And in both cases, when oxygen and nutrients diffuse from the inside to the tissues, this is down concentration gradient. And when carbon dioxide and waste substances diffuse from the cells to the blood, this is again down concentration gradient. Why after having, I might ask you, why after having received oxygen and nutrients, the cells give out carbon dioxide and waste? If I ask you such a question, then I'm asking you about cellular respiration and what occurs inside the cells. So this is the capillary tissue exchange. We have another type of exchange, and that other type of exchange occurs between the blood in the blood capillaries and between the air sacs. So oxygen will diffuse from the alveoli to the blood, whereas carbon dioxide at the venular end will diffuse from the blood to the air sacs, again down concentration gradient. Of course, here this is the air sac, and this is the blood inside the capillaries. Okay. Here there is something that is a bit more difficult than the rest. Notice that the blood that comes to the air sacs, see this deoxygenated blood that comes to the air sacs and that releases carbon dioxide or becomes unloaded from carbon dioxide. This is a branch of the pulmonary artery that comes from the right ventricle. And when the oxygen, when the blood becomes loaded with oxygen and becomes oxygenated and leaves the lungs, these are branches of the pulmonary veins that will pour that oxygenated blood into the left atrium. So let's talk about the purpose of the process of alveolo-capillary exchange. If I ever ask you about the purpose of the alveolo-capillary exchange, you would say it is to load the blood with oxygen and unload it from carbon dioxide at the level of the lungs. If I ever ask you about the importance of tissue-capillary exchange, you would say the purpose is to deliver oxygen and nutrients to the tissues and to receive carbon dioxide and waste substances from the tissues. Now, here we have a double pump. why do we need a double pump? First of all, what is a double pump? The heart is a pump, as we know. Why is the heart a pump? Because its wall consists of muscles, and these muscles can contract to pump blood. As you know, the left ventricle pumps blood. So we agreed upon this fact that the heart acts as a pump. What helps the heart act as a pump? That its wall is muscular. Why do we need a pump? In order to push or propel the blood into the arteries. But why do we consider the heart a double pump, and why do we need a double pump? First of all, why do we consider the heart a double pump? This is because the heart is two-sided. We have a half, the right half, as you see here, the right half of the patient, of course, not your right hand, that contains deoxygenated blood, and a left half that contains oxygenated blood. And it's not allowed for the two sides to mix up their blood. So here we have between the right atrium that contains deoxygenated blood and the left atrium, we have the interatrial septum to prevent mixing up. And between the two ventricles, we have the interventricular septum. And this is how we would have revised the importance of the two septa. So the heart is a double pump because we have a right side of the heart that pumps that receives first deoxygenated blood from the superior and inferior vena cava, and then pumps it along the pulmonary artery to the lungs. But we also have a left side that receives oxygenated blood via the pulmonary veins by the left atrium, and then pumps this oxygenated blood into the aorta. Okay, so this double pump is essential because we have two circulations. We have the pulmonary circulation and we have the systemic circulation, and each of them has its importance that we will be discussing later on. Of course, it's extremely important to know the anatomy of the heart, first of all, in order to be able to describe correctly the functions of the chambers, and second, in order to be able to label a diagram of the heart, a blank diagram of the heart, if any, on the test. So we will start here. We have deoxygenated blood that comes from the head and neck. Therefore, this is the superior vena cava. And then we have deoxygenated blood that comes from the lower part of the body. And here we have the inferior vena cava. And here we have the right atrium. There we have the right ventricle. Between the right atrium and the right ventricle, we have the tricuspid valve. And this is the direction of flow. So it's a good opportunity to review the function of the superior vena cava, the inferior vena cava, and the right atrium. So if I ever ask you about the function of the superior vena cava, you would say that the superior vena cava carries deoxygenated blood from the head and neck to the right atrium. And if I ever ask you about the inferior vena cava, you would say that the inferior vena cava carries blood from the lower part of the body to the right atrium. And if I ever ask you about the function of any atrium, you would say receive and send. So if I ask you about the right atrium, you would say that the right atrium receives deoxygenated blood from the superior and inferior vena cava and sends it to the right ventricle. And it's a good opportunity again to review the function of the right ventricle. So the right ventricle's function is, again, to receive and send. So it receives deoxygenated blood from the right atrium and sends it through the pulmonary valve into the pulmonary artery. And now it's a good opportunity to talk about the valves. Here we have the tricuspid valve and we have the pulmonary valve. So what is the function of, first of all, what is the location, the site of the tricuspid valve, and then what is its function? So the location of the tricuspid valve is that it lies between the right atrium above and the right ventricle below. This is the location. What about the function of the valve? Any valve in the heart is a one-way door that allows forward flow and prevents backward flow. And by forward flow, I mean the correct flow, or the flow in the right direction. So here, the tricuspid valve allows the flow, and you have to mention the type of the blood that passes. So it allows the flow of deoxygenated blood from the right atrium to the right ventricle during ventricular diastole and prevents backflow during ventricular systole. So you have noticed that I said allowed, I said prevented, I said the type of blood, or I mentioned the type of blood, and I mentioned it closes during diastole or systole, and it opens during diastole or systole. I'll pause for a minute and then resume recording. Hello again. And now we're going to discuss the functions of the structures that are present on the left side of the heart. And we will be starting by the pulmonary veins. As you know, the pulmonary veins are the only veins in the human body that carry oxygenated blood in an adult person, of course. And the pulmonary veins, as you see, pour their oxygenated blood into the left atrium. So if I ever ask you about the pulmonary veins, the function of the pulmonary veins, it's more important to say that they carry oxygenated blood back to the left atrium from the lungs than saying, for example, that they are the only veins that carry oxygenated blood. Because if you say they are the only veins that carry oxygenated blood, then you would be just describing the function of the, just describing the pulmonary veins. Now you see that they are 4, 2 on each side, but those 2 that are on the right side coming from the right lung will not pour their oxygenated blood, of course, on the right side, but they will pass like this behind the heart and pour their blood in the left atrium. So if I ever ask you about the function of the left atrium, you would say the left atrium receives oxygenated blood via the pulmonary veins and passes it to the left ventricle. And if I ever ask you about the function of the left ventricle, it's again receives and pumps. So, and it's more important to say pump with the ventricle. With the atria, you can just say pass. It passes the blood. It's true that they contract and that they pump, but their pumping is much, much weaker than the pumping of the ventricles. So the left ventricle receives blood from oxygenated blood, of course, from the left atrium and pumps this oxygenated blood into the aorta. And then we have the aorta that carries the oxygenated blood to all parts of the human body. As you can see, the aorta is curved like this, and it has 3 major branches to supply the whole human body. It will divide into ascending branches to the head and neck and descending branches. Here, as you can see, between the left atrium and the left ventricle, we have the mitral valve or the bicuspid valve that is made only of 2 cusps, and it allows, as usual, if I ever ask you what's the function of the mitral valve, you would say that the mitral valve allows the passage of oxygenated blood from the left atrium to the left ventricle during diastole or during relaxation of the left ventricle and prevents backflow during left ventricular systole. Both the tricuspid valve and the mitral valve are called atrioventricular valves. So if I ask about the atrioventricular valves, then I am asking about the valves that lie between the atria and the ventricles. Then I'm asking about the tricuspid and mitral. But if I say the semi-lunar valves, then I would be asking about the pulmonary and the aortic valves. Now we come to the next question, which is the description of the function of each of the pulmonary valve and the aortic valve. Now, I'm talking about the normal pulmonary valve. The normal pulmonary valve will allow passage of deoxygenated blood from the right ventricle to the pulmonary artery. When can blood pass from the left ventricle to the from the right ventricle to the pulmonary artery? When the right ventricle contracts. And then it closes to prevent backflow during diastole of the right ventricle. On the other hand, if you are asked about the function of the aortic valve, you would say that the aortic valve allows, as you see the direction of the arrow, see how the blood flows from the right ventricle to the aorta. So the aorta has to allow this blood flow during left ventricular contraction from the left ventricle to the aorta. So it's open and then it closes in order to prevent the backflow of blood from the aorta into the left ventricle. Now I might ask you another question that is related to the valves, which is, what is a murmur? A murmur is an abnormal sound. When you put the stethoscope on the heart, on specific areas of the heart, you hear lub-dub, lub-dub, lub-dub. Lub is the first heart sound, and it is due to closure of the atrioventricular valves. They close together, they form like a snap sound, and you hear the first heart sound, which is lub. And then closure of the semilunar valves gives you S2, or the second heart sound. These are the normal heart sounds, and both are due to closure of valves. Maybe we haven't mentioned this in the classroom, but it's good to know that S1 is due to closure of the AV valves, and S2 is due to closure of the semilunar valves. Now, if the valves are normal, then the blood flow is regular and laminar, and there is no turbulence, and you hear no sound. But if the blood flow is non-laminar, non-regular, and turbulent, then you hear a murmur. So what is a murmur? A murmur is an abnormal heart sound that is superimposed on the first heart sound and on the second heart sound due to turbulence of blood. But this is not sufficient to define a murmur, because people might be asking themselves or wondering, why would the blood flow be turbulent? The blood flow is turbulent when the valve is narrow and stenotic, or when the valve is incompetent and cannot close properly. Let's take examples. Now, if we look at the famous mitral valve, at the famous mitral valve here, that lies between the left atrium and the left ventricle. We have two problems with the mitral valve that might cause diseases or symptoms. We have mitral stenosis and mitral incompetence. Let's first ask about mitral stenosis. What is mitral stenosis? Mitral stenosis is narrowing of the mitral valve, and in Egypt, this is most probably due to rheumatic fever. The second question would be, why is there a murmur in case of mitral stenosis, or justify the presence of a murmur in case of mitral stenosis? The justification is like this: when the mitral valve is narrow, then there is increased resistance to blood flow. So the blood passes through a narrow opening, or a narrow orifice, and this results in turbulence and results in a murmur. Let's take the other example, which is mitral incompetence. What is mitral incompetence? Mitral incompetence means that the mitral valve cannot close adequately, okay? If the mitral valve cannot close adequately, then what happens is that when the left ventricle contracts, part of the left ventricle contracts to push blood into the aorta, but part of the blood would backflow abnormally into the left atrium. So this is not a normal direction of flow. That's why it causes turbulence and it causes a murmur. There remains one important question about the timing of the murmur in case of mitral stenosis and in case of mitral incompetence. Is it a systolic murmur or a diastolic murmur? So you have to review your the hemodynamics of the heart. Now, if there is mitral stenosis, then the turbulence would occur during passage of blood from the left atrium to the left ventricle, which is at that time in diastole. So the murmur of mitral stenosis is a diastolic murmur. But if the mitral valve is incompetent, then the backflow would occur during systole. The left ventricle is contracting and the blood backflows into the left atrium, so it is a systolic murmur. What happens in mitral stenosis, and all the justifications we mentioned can be applied to the tricuspid valve. So in case of tricuspid stenosis, you will have a diastolic murmur for the same reasons. And in case of tricuspid incompetence, you would have a systolic murmur. There remains an important question about the influence of each of these valvular diseases on the lungs. Say, for example, we have tricuspid stenosis or even, mitral stenosis. Here, this is the mitral valve. Say we have mitral stenosis or we have mitral incompetence. In both cases, there will be back pressure on the left atrium and therefore back pressure on the pulmonary veins and therefore back pressure on the lungs. So the air sacs would be surrounded by congested blood capillaries. And if the blood capillaries are congested, then they will press on the air sacs. There will be diminished exchange and the blood will be less oxygenated, less loaded with oxygen, and less unloaded from carbon dioxide. That's why, because of this low oxygen in the tissues and this low capillary exchange, the patient will suffer from hypoxia. Tissue hypoxia, less oxygen will reach the tissues. I might also ask you about capillary congestion. What happens when there is congestion of the pulmonary capillaries? There are three main symptoms. The first symptom is dyspnea, which is just difficult breathing. The second symptom is orthopnea, which means that as soon as the patient lies down and puts his head on the pillow, he sits down immediately because he cannot breathe at ease. He feels distress, and this is the most serious condition, even more serious than paroxysmal nocturnal dyspnea, because orthopnea occurs throughout the day and the night, 24 hours over 24 hours. But paroxysmal nocturnal dyspnea occurs at night when you go to bed, you sleep for an hour or two, and then fluid gradually accumulates in your lungs because of the pulmonary congestion, and therefore you suddenly feel distressed and you sit down struggling to breathe and having fresh air, and you cough frothy sputum. This froth is from the fluid that accumulated in the lungs. Another thing is that when we have congestion, for example, let's talk about another cause for dyspnea in case of mitral stenosis. If you have mitral stenosis, another cause for dyspnea would be that there is resistance to blood flow, there isn't enough blood that reaches the left ventricle, there isn't enough blood that reaches the aorta, and therefore there isn't enough blood that reaches the tissues. Now we're going to talk about ventricular failure. So we can talk about left ventricular failure and right ventricular failure. Whether it is left ventricular failure or right ventricular failure in general means what? Ventricular failure in general means that the ventricle cannot pump adequate amounts of blood to the circulation. Now, if you have left ventricular failure, then the left ventricle cannot pump adequate amounts of blood to the aorta, and therefore the blood would accumulate in the left atrium, and there would be congestion of the pulmonary veins and the lungs. If you have right ventricular failure, I forgot to tell you something, that whenever you are required to mention the clinical picture of left ventricular failure, you have to divide it into two broad categories. The one that I have just mentioned, which is due to low cardiac output, and actually both are due to low cardiac output and pulmonary congestion, because when the left ventricle cannot pump blood adequately to the tissues, there will be pulmonary congestion. But the symptoms I've just mentioned, dyspnea, orthopnea, and paroxysmal nocturnal dyspnea, are symptoms due to pulmonary congestion and back pressure on the lungs. But there are other symptoms due to low cardiac output and due, of course, to low oxygen in the tissues, or tissue hypoxia, which are constant fatigue, constant dyspnea, and a constantly high rate of the heart. Now, if we have right ventricular failure, there will be again symptoms due to low pumping of blood into the pulmonary artery, and therefore low exchange, resulting in dyspnea. And there are symptoms due symptoms due to back pressure on the right atrium that are congested superior vena, a congested superior vena cava, and a congested inferior vena cava. A congested inferior vena cava will be manifested clinically or shown clinically as lower limb swelling or edema, whereas a congested superior vena cava will be manifested in the clinical case by congested internal jugular vein. The internal jugular, you can say jugular or jugular, both are correct. How can I know that I have congested neck veins? We put a horizontal level at the level of the sternal angle, and we seat the patient at 45 degrees with the chair, with the bed, or with the horizontal, and then we look at the upper border of the visible pulsations, and they are not felt, they are only seen, and you put another horizontal level, and in between the two horizontal levels, you measure the vertical distance, and that vertical distance should not be more than 2 to 3 centimeters. Okay. Now we have, by having said this, we would have covered many slides on the presentation, and of course the question about the septum is very important. We talked about the function. Sorry, I'll pause for a minute. We said that the function of the normal interatrial septum is to prevent mixing up of deoxygenated blood in the right atrium with oxygenated blood in the left atrium. And the function of the interventricular septum is to prevent mixing up of oxygenated blood on the left side, on the left ventricle, in the left ventricle with deoxygenated blood in the right ventricle. But sometimes there are congenital defects. So we have an atrial here, for example, you can see an atrial septal defect, but there could also be in another patient a ventricular septal defect. Here, for example, a defect means that the wall in between the two atria is not perfectly closed. It might close with age as the child grows up, but it might need surgical interference later on in life. If the defect is relatively big and it didn't close, then this would result in mixing up, and this would result in diminished oxygenation of the tissues and tissue hypoxia with all the symptoms of tissue hypoxia. Of course, it's needless to say that if this septum is if this defect is in the interventricular septum, the condition might be more serious. Why would it be more serious? Because the ventricles contain blood at a higher pressure and a larger volume of blood, and therefore the defect would be more serious. Of course, when you have tissue hypoxia, there is a response from the tissue. When the cells do not receive adequate amounts of oxygen, the heart should beat at a more rapid rate, should pump more blood, and the lungs should inflate and deflate at a more rapid rate. The lungs should inflate and deflate at a more rapid rate to enhance the process of exchange and oxygenation of the tissues and unloading them from carbon dioxide. The heart beats at a rapid rate at a more rapid rate to make up for the loss of oxygen. So this is an answer to why do we have an increased heart rate and an increased breathing rate when there is tissue hypoxia. Another important item is the coronary circulation. As you see, the coronary arteries lie on the surface of the heart, and they arise from the beginning of the aorta, where you have blood under the highest pressure, and the coronary arteries fill only during diastole, because during diastole the heart is relaxed and they have a wider diameter. The coronary arteries function. What is the function? How do we call first the arteries that supply the heart muscles or the myocardium with oxygen? We call them the coronary arteries. What do they do? What is their function? Their function is to supply the cardiac muscles or the myocardium with oxygen and with nutrients. Why are the coronary arteries easily obliterated or occluded? This is because they have a very small diameter. And how can they be occluded? How can coronary arteries be occluded? They can be occluded by two ways or via two processes. The first process is the deposition of fat or cholesterol on the inner lining of the vessels. And the second is a blood clot or a thrombus. This occlusion can be a partial occlusion causing angina pectoris or a complete occlusion causing myocardial infarction. Now, in angina pectoris, there is relative deficiency of blood reaching the myocardium via the coronary arteries. But you have to take into consideration that this relative deficiency of blood might be caused by a partial occlusion due to a thrombus or due to a deposition of cholesterol that causes narrowing of the coronary arteries, but it might also be due to hypotension, for example, diminished blood pressure, and therefore diminished blood flow to the myocardium. In such a case, the arteries might be fully open. There is no cholesterol deposited in the inner lining. There are no thrombi, no clots, but still there is relative insufficiency of the blood just because the perfusion or the flow of blood to the heart muscles is not adequate. So you might have a young person with no cholesterol deposits, with no thrombus, yet having angina and the pain of angina just because there is inadequate flow of blood to the tissues. Maybe this young person had an accident and bled or lost bled the past tense of bleeding, to bleed. He bled intensively, and therefore the volume of blood diminished, the flow of blood to the heart diminished. If you have relative insufficiency of blood that reaches the tissues, this means that there aren't adequate amounts of oxygen that reach your tissues, nor adequate amounts of glucose, and therefore your cardiac muscles start to form lactic acid, and this causes pain. The pain in cardiac disease, in coronary artery diseases in general, whether angina pectoris or myocardial infarction, is behind the sternum, retrosternal. When I say retrosternal, I mean behind the sternum, and it is a very peculiar type of pain. It's not dull, diffuse pain. It's not sharp stabbing pain. It's crushing pain or squeezing pain. The patient feels that's why people think they're gonna die, because there is something that squeezes them behind the sternum, and this is very characteristic pain. The sole difference between angina pectoris and myocardial infarction is that in myocardial infarction, you have complete obliteration of the artery. And here it has mostly to be due to occlusion by a thrombus or cholesterol deposit. So the condition is more severe. The pain is still retrosternal in myocardial infarction. The pain is still crushing and squeezing, but it is much, much, much more severe. The second difference, other than severity of the pain, is how the pain is relieved. In case of angina pectoris, the pain is relieved by rest and coronary dilators, whereas in myocardial infarction, it's not relieved by rest and coronary dilators, and you have to give a medication that dissolves the clot. The third difference is that in angina pectoris, there is no death of the cardiac muscles. Therefore, it is a reversible, less severe condition, whereas in myocardial infarction, because of the complete obliteration of the blood supply, there is death of the cardiac muscles in the area supplied by that branch of the coronary that is supplied. Of course, if all the coronaries are occluded, or if a major branch that supplies most of the heart, like 2/3 of the heart, is occluded, this will result in extensive death of the cardiac muscles that might lead to death of the patient because of left ventricular inadequacy and severe acute left ventricular failure. Of course, the differences and the similarities between angina pectoris and myocardial infarction are very important. We have already discussed the valves, but only one item remains, which is rheumatic fever. As you know, rheumatic fever is a condition where the patient presents with fever, bilateral knee swelling, redness, warmth, non-itchy rash, especially on the back. And this is mostly like between a week or two after an attack of sore throat for which he has received an incomplete dosage of antibiotic. And the arthritis is fleeting, meaning that after the arthritis, I mean the inflammation in the joint, is fleeting, because after having had bilateral pain swelling, the patient might have bilateral ankle swelling, and there are like small firm nodules under the skin that are very characteristic of rheumatic fever. If I ever ask you about the clinical picture, you would say this. But if I ask you about the pathogenesis of rheumatic fever, how rheumatic fever develops, you start by an incomplete dosage of antibiotic, incomplete killing of the streptococci, residual streptococcal antigens that stay in the tissues, attachment of these residual or remaining streptococcal antigens to your connective tissue cells, your connective tissue cells being recognized as foreign non-self cells by your immune system, and at the end of the day, your immune system attacking these cells with the antigens that they carry, resulting in release of chemicals that cause the inflammation, the swelling, and the redness, and all the symptomatology of the patient. And that's it. Thank you, guys. Of course, you're going to read the presentation, and if you have further questions, you might send me on WhatsApp. And I highly recommend that you review all the assignments, all the previous assignments, especially the feedback on the assignments, in order to avoid repeating your mistakes. Best of luck with your final test, inshallah, and then enjoying you the best of happiness and peace of mind during your summer vacation. And see you on Tuesday, inshallah, after the test.